Okay, so the next step, after we have done all these steps for all these controls and the strength of immunoprecipitation, is just to run MACS2. So let's go here to see if MACS2 is available, and MACS2 call peak. So MACS2 call peak, a peak is actually, yeah, let's maybe draw that. So we have the gene here, and we have the reads stacked here, and here in a second gene. So this makes a peak here and another peak here, and this peak here denotes that the transcription factor TAL1 is bound here, and here TAL1 is bound here. Okay, so MACS2 will try to infer these peaks here in order to denote where TAL1 may bind. Now, it's worthwhile actually to understand a little bit the logic behind MACS2. Okay, so let's suppose that this is the genome. We have the plus strand and the minus strand, and TAL1 is this gene and is located here. So let's suppose that this is TAL1. Now, since we have put an antibody here and we have precipitate this thing, and we have also used sonication to cut the DNA, perhaps we cut it here and we cut it here, which means that there is a fragment like this one. And what we are doing now is that we remove the TAL1 and we sequence this. And the way that we sequence is the usual way. We either sequence some 50 base pairs here, so we will sequence this part, or perhaps in another cell cut has been done here, so we sequence this part. In another cell cut has been done here, so we sequence this part, and so on. Which means that regarding the 5' end, we will either sequence somewhere here. So regarding the plus strand, we will always sequence somewhere here, and regarding the minus strand, we will always sequence somewhere here. Okay, so the point is the following. Now, if we will not have the same number of reads stacked in the plus and minus strand around the protein of interest. We'll have several reads on this side of the plus strand and several reads on this side of the minus strand. Which means that if we see the distribution of reads that we have here and here, it will form something like that. So these are the reads on this side, and these are the reads on this side. We have more reads around here for the plus strand and more reads around here for the negative strand. Now this length should be approximately the same as the fragment size. Because on average, most of the reads here will fall in some location, and fragment size units on the right, the reads from the negative strands will follow. Because what we sequence is actually the beginning of each fragment. You remember that if the fragment is like that, we sequence this part in the singlet end. And from the minus strand, we will sequence this part. Now it's possible that the fragment is bigger, so we'll sequence this part here, a bit on the right. And also the plus strand fragment might be bigger, so we'll sequence somewhere here. But on average, we will sequence, the reads that we will sequence from the plus strand and the minus strand will differ by the average fragment size, which will be this distance here. What MACS is doing is that exploits actually exactly this fact, that from the plus strand we expect some read somewhere here, and from the minus strand we expect to have more reads fragment size units away. So it tries, in the beginning it looks for regions that have this profile, it compares them to the input in order to be secured that we have more reads in the immunoprecipitated file compared to the input, and then it shifts these fragment size half units on the right and this fragment size half units on the left, and makes a big peak from these two guys, like this one. The other thing that we should pay attention is the following. We don't really care what's happening in each individual. So remember that in our experiment, we have two guys, we have two immunoprecipitated guys and two controls. So two different individuals that we have measured the immunoprecipitation. However, we don't really care about the differences between these two individuals, but what we try to infer is one single answer for where TAL1 binds on these mice. So what we should do eventually, this is these two mice, so mouse 1 and mouse 2, which is the TAL1 dataset and TAL2 dataset here, to merge them. So we'll merge them in order to have more reads and find more accurately the binding locations of the TAL1. Now this is also justified because if you remember, we had done this experiment here, and we had seen that, let me show that again, it's similar to this one, that there is correlation between the TAL1 guys. So this one, for example, like here. So it means that the two immunoprecipitated G1E cells behave similarly, so we will just merge them to increase our signal, to obtain more reads, in a sense. Okay, so we proceed with Galaxy and MACS2, Colpic. And are you pulling treatment files? The answer will be yes. We want to pull treatment files. Okay, and which are the treatment files? So we have, if I remember well, it's the 29 and 28. Do you have a control file? Yes. Are you pulling control files? Yes. So which are the control files? It's the 26 and 27. What we have is single-end BAM, so we leave this as this. The effective genome size is the genome that is mappable, because we have a genome that has different occupancies from heterochromatin. Not all the genome is mappable, but they have done previous experiments and they have calculated these things. So we choose here mus musculus, since we have a mus musculus file. And the next thing is Let me check. What kind of outputs we want to have for our results. So we go to additional outputs here. Where is the additional output? They are here actually, so I think I will just choose all of them. And I think we are okay to go. I will just click Run Tool. Things are going. Now the next step will be to evaluate these outputs and create some plots. So I will just stop here, and next time in the next video we'll just discuss about the output of MACS2.